Hi everyone. Today we will discuss meiosis, a special type of cell division in sexual reproducing organisms used to produce the gametes, such as sperm or egg cells. You may notice that I have combined the materials from different chapters, so please follow the pages for study. I already showed you these slides in last lecture. This is just a reminder that meiosis, one diploid cells will generate four haploid cells through two times of cell division. Now let's watch a video together.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
The video contains a lot of information. I would like to go through some important concepts in details. We have mentioned that meiosis produces 4 haploid cells, and each cell contains only 1 copy of either paternal or maternal chromosomes. Paternal means related to father, and maternal means relating to mother. And you should have learned these from genetic courses before. 1 cell to 4 cells, indicating that 2 rounds of division happened. The first cell division is named meiosis 1, and the second cell division is named as meiosis 2. You need to remember the common and different features between mitosis and meiosis are always the questions shown in exams, including MCAT. Okay, let's go through this process quickly with figures. So this picture shows the stages of meiosis 1. First, you can see that this first division follows the name PMAT, this phase pattern. Importantly, you can see the X shape of chromosome remains after division, meaning the sister chromatids do not separate during meiosis 1. Okay, another important point is the bold words shown here, showing that the homologous chromosomes pair with each other in meiosis 1. So what is the purpose of pairing? Yes, crossover can happen to generate new combinations, which are responsible for genetic variations in offsprings. In meiosis 2, the second cell division, again, PMAT, still 4 stages. For meiosis 2, the cells will not make another copy of DNA, so no DNA replication. Remember, we mentioned that X-shaped chromosome remains after meiosis 1. In meiosis 2, the 2 chromatids were separate, and X-shaped will not become now it's become like an I now. So the division is quite like a mitosis. But because there's no DNA replication, the genetic materials cut half. So at the end, 4 haploid cells will be generated. 1, 2, 3, 4. So this slide is very important because I bolded every single word, but just kidding. This is important because it summarizes the common feature and the differences between mitosis and meiosis. We have gone through this, so I will not repeat. Here, because meiosis generates genetic variation, I'll spend some time to explain how does this happen. So there are 2 ways to generate a genetic variation. So one is meiotic crossover, and also the other one is random assortment of chromosomes. Let's first take a look at the chromosome movement during meiosis to identify when crossover happens. This picture shows a pair of homologous chromosomes, one paternal and one maternal. Okay. After DNA replication, each of them has 2 chromatids named sister chromatids. You need to understand that all these words mean. So we just learned that during meiosis 1, the homologous chromosomes pair and then separated to daughter cells. And the sister chromatids remain staying together until meiosis 2, right? So then the question is, why the homologous chromosomes need to pair together? It's just like 2 people meet and leave. They need to talk or do something together. It is hard to imagine that 2 people meet without saying anything and then just leave, right? So same thing here. The homologous chromosomes exchange genetic materials during their pairing. This is meiotic crossover. Let's see. So this picture shows how crossover occur. First, forget about all the labels, okay? Focus on the picture. What the picture shows you is all about chromosomes exchange their arms, right? So you give me something and I give you something back. It is quite fair. But several things I need to point out. So first, the crossover only takes place between the non-sister chromatids, which makes sense because people do not exchange things with themselves, right? Second, after exchange, new combinations are generated, all right? So that is the reason why siblings are not always the same. If you could understand the allele information, great. If not, we were getting to the gene and alleles later on. Okay, so all these patterns allele, we will study that later. So another way of generating genetic variation is called a random assortment of chromosomes, meaning distribute the chromosome into daughter cells randomly. Based on this picture, can you tell when the random assortment happens? Is it in meiosis 1 or meiosis 2? You can pause here to think because this one does not have a label, okay? So the answer is in the meiosis 1. You can see that all the combination and the assortment are in the top pictures. It's in these circled cells. That is in the meiosis 1, okay? In meiosis 2, just the sister chromatids separates. So, and there is no sorting happening. Now we are going to switch the gear and talk about something more directly related to our health. We know that the purpose of meiosis is to generate germ cells. The mature haploid male or female germ cell, also called a gametes, okay? We all know that a human has 2 kinds of gametes, sperm and egg. The process to produce sperm in male is called a spermatogenesis. And the organ of spermatogenesis is testis. In contrast, the process to produce oocytes in female is called oogenesis, and the organ of oogenesis is ovary. Although sperm and egg are both germ cells and need to go through meiosis, the spermatogenesis and oogenesis are quite different in timing and functionality of daughter cells. We will discuss this in more detail when we introduce the embryonic development chapter. Here, I would like you to remember several interesting facts, which can actually explain a lot of phenomena we observe in our daily life. First, sperm are continuously produced from puberty until death, and each primary spermatocytes, also known as the sperm cell before meiosis 1, they can produce full mature sperm. This is probably the reason why sometimes you hear that a 60-year-old man can have a baby, although it is probably quite rare. In contrast, all primary oocytes have already generated during the embryonic development of this female, and all the oocytes are arrested in meiosis 1 stage until ovulation. Okay, the meiosis 2 happens only after fertilization, and more importantly, only one mature ovum, that's also called an egg, can be produced from one primary oocyte. And why is that? Let's first take a look at a picture shows a female's biology clock. So the y-axis shows the follicle number because one follicle contains one egg. So this basically refers to the number of eggs. As you can see that the female has the most eggs before the female is born, and starting from puberty to 35 years old, the numbers of follicles still remain at a relatively constant level. Okay. However, after 35 years old, the numbers of follicles have significant reduction. After 45, the number is so low that it's very difficult for the female to become pregnant. Okay. Please keep in mind that no new primary oocytes can be generated after the baby girl is born. So now let's take a look at a picture to explain the differences between spermatogenesis and oogenesis better. On the left shows the spermatogenesis, which generates 4 sperms, as we expected, similar to the meiosis we introduced earlier. Before meiosis 1, it is the primary spermatocytes, and before meiosis 2 is the secondary spermatocytes. And after meiosis 2, the sperm cells are called spermatids. Okay, and then they were further developed to mature sperm with this tadpole shape. Things are different for oocytes. As we mentioned, the primary oocytes arrest in meiosis 1 until ovulation happens. Okay. Females ovulate once a month. And then that is the best time to produce a baby, all right? So interestingly, ovulation also completes meiosis 1, okay? And this cell division is not even. It produces one secondary oocyte and also a small cell named the polar body, okay? Polar body still carries half of the chromosomes, but it barely has any cytosol and organelles, and does not have the ability to be fertilized, okay? The polar body from meiosis 1 is also called a first polar body, okay? Then the meiosis 2 is initiated when sperm penetrates the second oocyte during fertilization. Again, another uneven division occurs, and this time the secondary oocyte produces one egg and another polar body, while the first polar body also divides into 2 daughter polar bodies, although they are tiny. So therefore, upon completion of meiosis 2, there will be 1 egg and 3 polar bodies. Sometimes we say these are secondary polar bodies. The polar bodies will die eventually, and then there's only 1 egg. So this is a comparison of the duration of meiosis in males and females, okay? As I mentioned, the spermatogenesis begins at puberty, but oogenesis begins during embryogenesis, but ovulation begins at puberty, right? So therefore, you can conclude that total time for spermatogenesis is approximately 64 days to generate a mature sperm, but the whole process for female is 12 to 50 years, depending on how soon the egg can be fertilized and form a zygote, all right? So zygote means a fertilized egg. Since we have talked about the reproductivity, I would like to introduce some technologies in this field. You probably heard that the world's first test tube baby, her name is Louise Brown, and she was born in 1978. That was a huge event, and many media have reported this. I show some of those here. Today, I'll show you with you how test tube babies are made these days. First, let's try to understand why we need to make test tube babies. When a couple get married, they would like to have their own kids. However, in average, 1 in 6 couples can have problem producing babies. We call this infertile, okay? A successful reproduction requires many factors, including healthy gametes, a normal oviduct, which is the place for fertilization, and a healthy uterus. Okay? Doctors have defined infertility as 2 categories. If this couple have trouble conceiving a first child, that's primary infertility. If they had a child before and have trouble conceiving a second child, then that's secondary infertility. I was wondering if there's anything like third or fourth infertility, but I Googled it, but didn't find such words, okay? So only primary and secondary infertility is referred. This slide shows you the major cause of a female infertility. Although the oogenesis is rather complicated, the major causes of female infertility is quite simple. So the main causes is the abnormal structure of the reproductive organs, such as oviduct, uterine, and ovaries. Cervical problems that may block semen may be another reason. Hormones related to ovulation problems, such as they call the PCOS, that means polycystic ovary syndrome. And PCOS patients may develop fluid follicles in the ovaries, which fail to regularly releasing eggs, and then that can cause infertility as well. Aging is a natural reason for female infertility. Before 35, there is not much of concern, but after 40 years old, the infertility increases very quickly. This is inconsistent with the decreasing numbers of follicles with aging. I don't know if you have ever thought about that, but in my opinion, evolutionarily, the purpose of animal life is to produce offsprings, right? So allowing these species to live on Earth for a long time. So some animals die shortly after giving birth, like certain species of fish and octopus. It is okay because they already finished passing their genetics, right? So, but humans are not animals. They are much smarter and find out different ways to extend their lifespan. So currently in the US, the average female lifespan is 81 years old, while back to 1900, it was 50, okay? In 1850, it was 42. You can tell in the early days, the lifespan of a human being was just long enough to finish birth and raising the children. So in contrast to lifespan, our reproductive system has not evolved that fast. not only the infertility, but also complications can occur when women get pregnant late. So we'll go through these issues at molecular levels in the future. These are the major causes of male infertility. We can see that anything that affects the quality, quantity, and the delivery of sperm would cause infertility or decrease fertility, which completely makes sense. For instance, we learned that testis is the organ for sperm production, and normally a testicle moved into the bag of skin hung below the scrotum before birth. However, undescended testicles haven't moved into its proper position, and they stay inside the abdomen because sperm development needs slightly cooler than normal body temperature. Undescended testes can result in low sperm counts, poor sperm quality, and decreased fertility. It has been documented that men with an undescended testicle have an increased risk of testicular cancer as well. So you can also see that varicocele is the main reason to cause male infertility, showing 37%. I would like to spend more time introducing varicocele next. A varicocele is an enlargement of the veins within the loose bag of skin that holds the testicles or scrotum. Varicocele is similar to a varicose vein you might see in the leg and may develop as a result of poorly functioning valves that are normally found in veins. In other cases, it may occur from compression of a vein by a nearby structure. Varicoceles often produce no symptoms but can cause low sperm quality, leading to infertility. The good news is that if symptoms raised, varicocele is treatable and most cases can be corrected by surgeries. This problem is very common and has more than 3 million cases in the United States every single year. Right? Due to these many cases of infertility, many assisted reproductive technologies, also named as ART, have been developed to expand the childbearing options. Here I name a few, okay? These include intrauterine insemination with donor sperm. As the name indicates, this technology involves placing sperm inside a woman's uterus to facilitate the fertilization. The goal of IUI is to increase the number of sperm that reach the oviduct and subsequently increase the chance of fertilization. Eggs can also be retrieved and donated. Under this procedure, the female donors first need to take hormones to stimulate the oocytes to become mature, which is called the ovarian hyperstimulation. When the eggs are fully mature, but just prior to rupture of the follicles, egg retrieval is done under the guidance of an ultrasound. In this way, multiple eggs can be obtained rather than one egg per month that happens naturally. Okay, this technology has to be combined with in vitro fertilization to generate babies. I'll introduce that in detail in next slide. Finally, called surrogacy. So this means one woman helps another woman to carry and give birth to a baby. So this is a controversial option and is banned in some countries, but it's available in Texas. And the current cost of a surrogacy journey in Texas is between $100,000 and $125,000. There are few insurance companies cover the cost of surrogacy, but the plan is extremely expensive. Okay, so this slide shows you how the in vitro fertilization is performed in labs. First, the gametes are retrieved, right? So both sperm and eggs gametes, they are placed in a culture dish when this fertilization can occur. The fertilized egg is also called a zygote. So the zygote are cultured in an incubator to let them grow until they become an early embryo before they can be transferred to the uterus. Then embryos can implant it to the wall of the uterus and continue to grow to a fetus. However, not every embryo can successfully implant it. Therefore, doctors usually transfer more than one embryos to increase the chance of pregnancy. So the major risk of IVF is multiple birth, okay? So a pregnancy with multiple fetus carrying the higher risk of early labor and low birth weight than like just a single fetus of pregnancy, right? So interestingly, IVF treatments are not as common as you would think. In fact, only about like 5% of couples who experience infertility actually move forward with IVF. Since the first IVF baby was born, this technology has been improved so much that now it is very safe and affordable. Currently, almost 2% of US-born babies being conceived with assisted reproductive technology, including some celebrities, such as one of my favorite singer, Celine Dion. Okay. There are also quite a few clinics that are specialized on assisted reproductive technologies in Houston. I searched fertility clinics in Houston, and Google pops up this map. You can see that most clinics are located along in the TMC region, but I have a friend working in one of those clinics. This can be a job option for biology major too, okay? The development of in vitro fertilization also makes early genetic testing possible. From the first lecture, we learned that we can do genetic testing using collected saliva. But doing so, we could tell the risk of development of certain diseases. So precaution can be applied prior to the onset of these diseases. The test can also be done at a prenatal stage, meaning it can identify whether an embryo or fetus has inherited a genetic disease or not. Our textbook has listed some metabolic diseases and birth defects that can be diagnosed by prenatal testing. We will go through many of them in details in the future. However, if the individual already carries a germline mutation, it is hard to change when the fetus forms or once the baby is born, right? So now with IVF and genetic testing at a very early stage, we might be able to change this situation. I'll give you an example. We talked about RB gene and the loss of heterozygosity. And one copy of RB gene is defective in all the cells that baby has, right? So the right copy, the other copy is the normal, but then this one is a defective, right? Say this person is lucky enough that the LOH never happens, okay? And he or she never develops retinoblastoma. However, this person does not want to pass this defective allele of RB to his or her babies. Okay, do you think this can be controlled? Now the answer is yes, but needs the in vitro fertilization to help. Remember, there is one copy of defective chromosome 13 showing as this blue here, but then the other copy is normal, all right? So when the person generates gametes, these two copies of chromosome 13 can segregate and distribute it to the two different germ cells, right? if the normal copy is chosen, then the offspring will not have a defected RB gene. So, and then if you can get rid of this one, right? So this can be done with a technology called pre-implantational genetic diagnosis, or named PGD. Okay, so this slide shows you how PGD is performed on an in vitro fertilized embryo. So this procedure is done under a specialized microscope. First, a single cell needed to be removed from a day 3 embryo for genetic analysis. On this left, okay, on the left, this is a holder to stabilize the embryo. On the right is a very, very fine needle that can suck one cell from the embryo. Removal one cell at the early stage of embryonic development won't affect the embryonic development. But the DNA from this single cell is enough for genetic analysis. If the cell has defective gene, it won't be chosen for implantation, this embryo, right? I think PGD is already very fancy because technically it is quite challenging to handle. But scientists are very smart and take advantage of a polar body. We learned that, right? So which appear to be not so important. We learned that when ovulation happens, it is the stage of secondary oocyte, right? And it contains one polar body with a completely copy of chromosomes. So this picture shows that the polar body can be removed from genetic analysis. Therefore, this is a genetic testing before fertilization. Is science pretty amazing? Okay, so these are the course content for today. Now I would like to spend a few minutes to practice a few questions together with you. So these are true or false questions. Let's see, the first one. Mitotic divisions reduce the number of chromosomes found in daughter cells. Is this true or false? So this is the false. So mitotic division do not change the number of chromosomes because DNA has been replicated. The second one, cytokinesis usually occurs just prior to mitosis. Is this true or false? So it's also false because cytokinesis separates the cells to two daughter cells. So it happens right after the mitosis. Okay, the third one, crossover is partially responsible for our genetic diversity. Is that correct? So the answer is true. Indeed, crossover exchanges genetic material and generate genetic diversity. The fourth one, random assortment occurs between chromatids of homologous chromosome pairs. True or false? So the answer is false. So this is the definition of crossover, not random assortment, okay? So the fifth one, a polar body once formed has no further function and dies. True or false? The answer is true. At least at present, no other function of polar body has been identified. Maybe in the future there's new discovery find out, but at this moment, the answer is true. Okay, the last one. Primary oocytes are haploid cells. Is it true or false? The answer is false. Primary oocytes haven't started division, therefore they are diploid cells, okay? Okay, let's also to look at several multiple choice questions, okay? The first one, in the cell cycle, the G1 phase represents what? I give you some time. You can also pause here for thinking. Once you have an answer, you can continue. So the answer is C here, okay? G1 phase represents a period of growth, not the others. A is the S phase, synthesis. B is apparently cell division is mitosis. And actual cell division, also mitosis, a stage just prior to meiosis, has nothing to do with the G1 phase. We'll talk about G1 phase is in the mitosis, not meiosis, right? Second, in an organism where 2n equals 8, how many chromatids will be presented in a cell that at the beginning of meiosis 2? You have all kinds of numbers. Think about it. I encourage you to pause here because this question is not, needs to think. Okay, so the correct answer is D. Because this question is asking chromatids, okay, then we only focus on chromatids, nothing else. You think about chromatids only, okay? 2n equals 8, meaning normally you have 8 chromatids, right? For meiosis, DNA replication will double the number, and before meiosis 1, you should have 16 chromatids, right? So after meiosis 1, 2 cells generated, and each should have half. So that means 8 chromatids. Since no more DNA replication happened at the beginning of meiosis 2, you should have 8 chromatids. So this can be confusing, but if you are clear on what the question asks and focus on it, then you will get it correct, okay? Let's see next. A cell in G0 state. I give you some time to look at the question. Okay, the correct answer is E. G0 phase has entered resting stage and not actively dividing. That's exactly the definition we talk about the G0, okay? So first of all, it's actively growing before cell division begins. So that's apparently not correct. Has a cleavage follow, this is division, no. So actively replicating chromosome S phase. It's in cytokinesis because G0 state is not dividing, so there's no cytokinesis happen at all. So this correct answer is only E, all right? Let's see, last one. What is performed to detect genetic disorders and birth defects in the fetus, all right? Detect a genetic disorder. What is this? So apparently this is B, prenatal testing. So this is pretty straightforward. Carrier testing, and then genetic screening, gene therapy, and then this. So like a genetic screening somehow is related, but not a, I would say prenatal testing is the best one before, because this is in a fetus, okay, prenatal testing. Genetic screening usually meaning it's including both prenatal and after people's birth, you can do all kinds of screening, right? So that probably the best answer. All right, so that's all for today. I'll see you next time.